Right, so global health and emerging infections, lecture number 3, new foes. So this is part of this series of lectures looking at global health and emerging infections, and we're going to look today at 3 case studies: SARS, pandemic flu, and the German STEC outbreak of earlier this year. Before we start, we need to actually look at the definition of emerging infectious diseases. What are these? This is actually very much a buzzword that people throw around in papers and probably in grant proposals as well, because it's a very sexy kind of thing to be working on. So in a simpler sense, we're looking at diseases that are caused by a newly identified species or strain, where this has just emerged into the medical field, into public consciousness from a blue sky, if you like, from completely unknown. Then we get new infections resulting from variants of existing organisms. So we know about influenza, but when we get a new kind of influenza coming along, we can get pandemic flu. Sometimes infections can spread from outside of their normal range to completely new areas. And you may have heard of West Nile virus, which has spread from the old world to the new world in the last decade or so. This is where it starts to, the people are starting to kind of stretch the use of the term. They talk about re-emerging infections, where we get diseases which we kind of know about, but they are kind of resurging in certain areas. Tuberculosis, drug-resistant tuberculosis, cholera in Haiti, for example. Now, there are hundreds of diseases that have been called emerging infectious diseases. In fact, there's a link here which takes you to the online supplement to a recent Nature article where they have I think it's over 500 of these diseases listed. We can't cover all of that, so I've just taken 3 examples to talk about today. Before we move on to those, just one more point. What actually causes emergence of new diseases? And there are a whole range of things here which can contribute. So human behaviour, human demographics, population pressures, movement of peoples, air travel, the easy way in which we can just move from one city to another thousands of miles apart in a few hours. Changing human susceptibilities, so the arrival of HIV and AIDS in particular meant that humans became a new niche for many other bacteria and fungi and so on that would not normally cause disease in us. Similarly, cytotoxic agents that people use when they're having transplants or treating cancers, these suppress our immunity and make us susceptible to things that we wouldn't otherwise be susceptible to. Another area of great interest at the moment is the potential effects of climate change on the patterns of disease, infectious disease across the world. Could we have malaria coming back to the UK if we start to get more clement winters? Economic development, land use, this is a big issue, driving animals out of their natural habitats into close association with humans, people having to use bushmeat as food. The bacteria, fungi, viruses change as well and adapt and become, you get new variants emerging. Get breakdown of public health, abnormal natural occurrences, so if we have earthquakes, tsunamis, those kind of things. And of course the influence of war associated with disease, and the risk of bioterrorism. So you've probably heard about the release of anthrax into the American postal system. There is a concern that this kind of thing might happen again in the future. Okay, just to kind of get you warmed up, let me just show you this trailer.
Groundbreaking ceremony for a new factory.
Did you mention seeing anyone who was sick? Anyone on a plane at the airport?
No, she said she was jet lagged.
The average person touches their face 3 to 5 times every waking minute. In between, we're touching doorknobs, water fountains, and each other.
Okay. No, go up to your room, honey. So we have a virus, no treatment protocol, and no vaccine this time.
You had a seizure this morning, Beth.
Do you have a history of seizures? Allergies?
As of last night, there were 32 cases.
Unfortunately, she did die.
Right. So can I go talk to her?
Mr. Omoff, your wife is dead.
What are you talking about? What happened to her? What happened to her?
Is there any way someone could weaponize the bird flu? Is that what we're looking at?
Someone doesn't have to weaponize the bird flu. The birds are doing that.
Watch this. It's transmission, so we just need to know which direction.
On day 1, there were 2 people, and then 4, and then 16. In 3 months, it's 1 billion. That's where we're headed.
They're calling out the National Guard. They're moving the president underground.
People will panic. It will tip over. The truth is being kept from the world.
Cook your samples, destroy everything. Hello. I need you to get me the names of everyone who serviced this room. It's an emergency.
You can't panic now.
I know.
I'm gonna get you home. I got people too, Dr. Cheever. We all do.
Don't talk to anyone. Don't touch anyone. Stay away from other people.
We're not sick!
It's figuring us out faster than we're figuring it out. It's mutating.
Has anyone seen the film yet? It's on in the cinemas now. Should be required viewing for this particular module. Now, the question is it plausible? Is there anything like that? Well, it's actually based on 2 diseases. One is SARS, which I'm going to talk about now. And in terms of the epidemiology, the film mirrors SARS. In terms of the symptomatology, it's more similar to a disease called Nipah virus, which some of you will be dealing with in your presentations. So let's talk about SARS. So what was SARS? SARS, the first pandemic of the new millennium, and started in November 2002. So there were some initial cases in southern China. In fact, the Chinese authorities tried to kind of cover it up or ignore it. Quite slow in reporting problems, and in fact the People's Republic of China did actually issue an apology later on for this. But by February 2003, there had been an outbreak in Guangzhou in the hospitals there involving patients, and also it spread to healthcare workers. There are cumulative over 300 cases, 100 of them in healthcare workers, and 5 deaths. And these were from some unknown acute respiratory syndrome. So people were getting something wrong with their lungs. Some of them were dying. Things start to get worse in terms of the global epidemiology in February, because a 65-year-old professor of nephrology from Guangdong starts to fall unwell, and he goes to this hotel here, the Metropole Hotel in Kowloon on the mainland, just opposite Hong Kong Island, and he stays there, and while he's there he infects 17 residents at that hotel, and then on the 22nd he's actually admitted to hospital. So he's there one day, but that one day was crucial in infecting 17 people. The contacts who were exposed to infection in Hotel M in the Metropole then travel on to Hanoi, Singapore and Toronto, starting new outbreaks in those cities. One of them, a US businessman, Johnny Chen, was a contact of Hotel M. He falls ill on the flight to Singapore, and he's admitted to hospital in Hanoi, and then dies in Hanoi. Another Hotel M contact then goes to a hospital in Hong Kong, starts a hospital outbreak in Hong Kong, and yet another contact goes to Toronto, and dies in Toronto. 5 family members are affected in Toronto. So you can see this dramatic spread within just a few days few hours, in fact, of the individuals and a few days before you get secondary cases around the world. One of the most poignant stories to come out of the SARS outbreak was the role of this guy, Carlo Urbani, who was an Italian doctor, worked for the WHO in Hanoi, and there was an outbreak in the hospital in Hanoi, an explosive outbreak, and he was the one that tipped off the world. He was the one that told the WHO something is happening here, something new, something frightening. And his descriptions of these cases alerts the health authorities around the world, and the race is then on to identify the virus. So he played a pivotal role here in actually shouting loudly and bringing this to the world's attention. Sadly, after seeing his first case, a month after seeing his first case, and 18 days after falling in on a plane to Bangkok, he actually died himself from SARS. So those of you who watch the film Contagion, not giving too much away, one of the healthcare workers there dies, and she's loosely based on Carlo Urbani as one of the kind of heroic cases among healthcare workers. He actually, in line with his kind of heroic status, he actually said here, if he could not work in such situations, what's he here for? Just answering emails, going to cocktail parties and pushing paper. He was someone who actually got out and saw the patients, described them, and sadly died. Anyway, on the back of that, the WHO then issued a global alert, and the name Southern Acute Respiratory Syndrome, or SARS, was coined, and there was a call for a global collaborative research effort. 21st of March in Hong Kong, the scientists there isolate a virus from an open lung biopsy. Turned out to be an unexpected kind of virus. It was a coronavirus, of which we'll say more later, and this was then confirmed in the US and in Germany. On the 12th of April, genome sequence from the University of British Columbia in Canada showed that this virus is distinct from all known human pathogens. Turns out that a virus almost identical to this SARS coronavirus was then isolated from palm civets, which is kind of animal here, you can see, and other game food animals. That was in June of 2003. And the epidemic was sputtering on, but then by July, the last cases of human-to-human transmission were occurring in Taiwan, and once those were over, the outbreak was effectively over, apart from the fact that there was some lab-acquired infection in Singapore in 2003. There was also some in Beijing and in Taiwan over the coming year. There were 5 new cases from December through to January of 2003 to 4 from animal to human transmission in Guangzhou. People were looking out for it and found it. And then in 2005, another source, probably the ultimate source of infection, was discovered, the horseshoe bat. And this is now thought to be the largest reservoir of infection in the wild. So it wasn't just those 3 cities I mentioned that got affected by SARS. It actually spread to a large number of countries around the world, and you can see the cases here in Europe, in Brazil, the USA, and even in the Russian Federation there, but obviously most of the cases here in China as the predominant sort of place where infection occurred. Perhaps more worrying than just where it was the fact that in lots of different places you got actual local transmission from person to person. So it wasn't that just people were getting on planes and then dropping dead, they were getting on planes and infecting someone, and then that person may have infected someone else. So in Toronto, Ottawa, San Francisco, Ulaanbaatar in Mongolia, Manila in the Philippines, Singapore, Taiwan, Hanoi and Hong Kong, all of those places you saw local transmission. And within the People's Republic of China, it spread to Guangdong, Jilin, Hebei, Hubei, Shanxi, Jiangsu, Shanxi, Tianjin, and in Mongolia. So pretty much all of China was affected. So what was the culprit? The culprit was this thing, SARS coronavirus, or SARS-CoV, Coronaviruses before this were thought to just be a kind of benign group of viruses. About 15% of cases of the common cold are caused by coronaviruses. This is a large, enveloped, plus single-stranded RNA virus in the jargon. It's got these irregular shape with club-shaped spikes all over it. The genome encodes a replicase and various structural proteins for the spike, envelope, membrane, and nucleocapsid. And the S protein, the spike protein, binds to a particular host receptor, angiotensin converting enzyme 2, or ACE2. And it's clear that actually mutations in that S protein occurred during the emergence of this as a human infection and during the epidemic. And these amount to adaptive changes, so that the spike protein was becoming more and more adapted to have a higher affinity for the human ACE2. And also looking at mutations in these genes, you can actually track the spread, the phylogeny of the outbreak as well. So we can look at changes that are actually associated with improved fitness of the virus, but also the changes which are just arising by chance and not having an effect on phenotype can be informative as well. So what is SARS in terms of its clinical picture? So the case definition, well, we have an incubation period of 2 to 14 days. Flu-like symptoms, nothing very specific there. High fever, myalgia, which is just the jargon word for muscle pains, lethargy, gastrointestinal symptoms, cough, sore throat, shortness of breath. So you get those things, and then to fulfill the case definition, you have to have had less than 10 days before the onset of symptoms, close contact with probable or suspected SARS patient, or during the outbreak, of course, they also just had to say, well, if you've been in an area with transmission of SARS, that's good enough. Most of the patients, vast majority of patients, have abnormal chest X-rays. So you can get these changes you can see here, this kind of opacity sort of across the lung there. In fact, now since the outbreak, we now have laboratory confirmed SARS, if you like. We can actually do laboratory tests. But at the time it appeared, obviously, that wasn't possible because we didn't know what the virus was, didn't have any reagents to test for in the laboratory. We know that it's human-to-human transmission is happening through direct or indirect contact of mucosal surfaces with infectious respiratory droplets or with fomites. So fomites, if you look at the reviews of that film Contagion, people say, oh, I learnt what the word fomite means. Fomite is just a general word for anything you might kind of touch. So things like your pen, books, toys, anything where you can actually get transmission from that object to a human. Survives pretty well outside the body, 2 to 3 days on dry surfaces. And in Hong Kong, they actually had this explosive outbreak in a particular housing estate, the Amoy Garden housing estate. And what happened there was that the traps in the drains, they dried out. These U-tubes, U-bends, they dried out in those sewage drains, and then these exhaust fans generated an aerosol in the toilets, and the aerosol then went up through these the kind of light wells that were connecting different floors, so it spread up the buildings. It's also now known that there are 5 different flights on which SARS was transmitted within the commercial aircraft setting. So again, amazing potential there for spread. So we now fairly confident that this is a cross-species jump from civets and or horseshoe bats to humans. Now the second case that was described was a chef with multiple animal contact. I don't think I'm being too outrageous by saying that people in southern China, particularly in Guangzhou, will eat just about anything they say. If it flies, they'll eat it. If it swims, they'll eat it. If it crawls, they'll eat it. And so there is a great taste for exotic animals there, or what we would say exotics like civets and so forth. I don't know if people actually eat bats. Anyway, as I said a couple of slides ago, you can actually draw family trees, phylogenies, and here's one looking at the broad picture, looking at lots of animal and human coronaviruses, and you can see here the human SARS coronavirus is very, very tightly linked to the civet and the bat SARS coronaviruses there, and so this forms a cluster within coronaviruses. How did they control it? Well, they had to break the transmission, chain of transmission from infected to healthy person. Case definition and detection, prompt isolation and contact tracing were all very important. Treatment with interferon was given, steroids, protease inhibitors were given with ribavirin, and convalescent plasma was used that contained neutralizing antibody. There was this creation of an emergency operating centre, institutional support, so efficient quarantine was implemented in Hong Kong, Canada, Singapore, Taiwan. Schools were closed down in Hong Kong and Singapore, and in these situations legislation was brought in. The WHO played a role in facilitating international collaboration and control, so there were travel alerts and restrictions. One of the most controversial decisions during the outbreak was that the WHO effectively put the whole of Toronto into quarantine, suggested that only essential visits to Toronto should be encouraged. There was also screening of airline passengers using thermal imaging, so you'd walk through and they would get an image of you, and if you shone out, looking as if you had a fever, they'd pull you to one side and you'd be screened. So a massive international collaboration. So after the outbreak, what were we left with? Well, 37 countries around the world, over 8,000 cases, 774 deaths. So a case fatality ratio about 1 in 10, about 10%. If you get old and you get SARS, you don't fare quite so well. 50% fatality in the over 65s. Where did it come from? Why did it happen? Well, part of it was the rapid economic growth in China. This is priming this demand for exotic food, animals such as civets. Overcrowded cages in the markets there, no biosecurity in the wet markets. And the fact that viruses can, on occasion, it's kind of an idiosyncrasy, if you like, serendipity, they just can jump from animals to humans on occasion. But then after it spread into the human population, it was then facilitated by this rapid global dissemination with international air travel in particular. And also in some of those hospitals in China and in Vietnam, people weren't as aware of hospital infection control as they might be in a more sophisticated setup in a richer country. Really, this was, it had an unparalleled dramatic impact on healthcare systems, economies, societies. And fortunately, we did manage to control it, but there is that concern that something like this could happen again, and will we be ready? If we get the same kind of conditions again, I mean, the reservoir still persists out there. Next time, we hope we can actually get there a bit quicker, treat perhaps. Maybe even vaccinate. There's vaccine research programs. There are now over 4,000 publications on SARS, but there's still plenty of gaps in our knowledge. So, we can't get too complacent. Now, I always like to give you a break every now and then. I'm now actually going to get a guest speaker from Canada. She's not actually going to come in person, but I've ripped this. I've managed to capture this video. This is Jennifer Gardy. And she's going to tell you about the experiences with swine flu in 2009 and how they differed from our experience with SARS. So let me just get Jennifer ready.
They were doing all right. So Richard's talk really brought up two things. It brought up that there's a lot of new technology that's out there, and there's a lot of new attitudes towards technology, and that public health in the 21st century really has to capitalize on this new technology and on these new attitudes to become basically public health 2.0, a new model for delivering public health services. So in listening to Richard's presentation, I was reminded of something very similar to this thought of this new model of delivery that I had written about H1N1 back in May. And so in my other life, when I'm not doing CDC-related things, I blog for the Globe and Mail's globecampus.ca website as Nerd Girl. It's a lovely photo of me with very good lighting. I don't normally look that good. And back in May, when swine flu first broke and appeared on people's radar, I wrote a post about some of the things that, as a scientist, I found really fascinating about the current outbreak. And one of those things was the idea that this H1N1 outbreak is really world's first open source outbreak. And what I meant by that was that data about this virus was being generated in real time, and it was being shared with researchers around the world freely with no restrictions, and the community had really come together as kind of a hive mind to solve this problem of pandemic influenza. So reminded of this idea that I had by Richard's talk, I figured that this would be my suitably TED-worthy topic. So what I'm going to talk about today is this idea of the open source outbreak, or outbreak 2.0. You know, what is it? What are the factors that have enabled it? And how did it really unfold with H1N1? And what might we see as far as outbreak 2.0 for the next outbreak? So I figure I'll give you my take-home point right now, no sense in waiting till the end, and that is basically that advances in technology, particularly around genome sequencing and just, you know, bioinformatics and infrastructure, have really contributed to this, as has a sea change in scientists' attitudes towards becoming more open access oriented and more collaborative. And really those two things together have brought us to the brink of this new era in public health, where our response to emerging infectious diseases and emerging threats is much more rapid than it's ever been in the past and much more collaborative than it's ever been. So we'll hop in our awesome time machine and go back with Doc to the heady, carefree days of March 2009, when swine flu was nary a blip on anybody's radar. I've got a calendar here. There's March, April, May, June, and mid-March that public health officials in Mexico started picking up increased flu activity. And this increased throughout late March and early April. And on April 15th, the first two cases of what would ultimately be recognized as H1N1 were picked up in California. And the screening program in California kind of triggered a warning bell, because these were two cases that didn't resemble any previous human influenza that we had seen before. So the Californian officials called in the CDC, and about 6 days later, the CDC was able to confirm that this was a novel swine origin influenza virus. So as soon as they issued this advisory, public health officials, basically the world over, started looking for this virus in their own population, and sure enough, they found it. They found it here in Canada, they found it elsewhere in the States, they found it in Europe, and by the time the 29th rolled around, just over a week later, it had basically been found everywhere. You know, over that week, the number of cases was pretty much doubling every day. So on the 29th, the World Health Organization raised the pandemic threat level to 5, meaning that a pandemic was imminent. And as we all know, the number of cases steadily increased throughout April and May, and finally on June 11th, a pandemic was officially declared by the World Health Organization. In Canada, as of June 11th, when the pandemic declared, at that point we had about 5,000 laboratory-confirmed cases of swine flu. And using a figure of 20 to 30 cases out there in the wild for every lab-confirmed case, that meant we were looking at about 100 to 150,000 people that were infected at the beginning of June. So where does the idea of open source outbreak fit in with all of this? Well, if we go... We'll go back to our calendar in a second second. I love this shot of the sneeze. It's so disgusting. It's the perfect public health picture. As people were sharing germs and this pandemic was spreading around the world, so too were researchers sharing data. And if we go back to the calendar, we can see that on April 25th, just this, like 4 days after the CDC confirmed this novel infectious agent, we already had the first genome sequence for swine flu, the blueprint for making the swine flu virus. And the very next day, this is so cool, a group of international researchers, there's 13 people representing 8 institutions across 4 countries, came together and decided that they were going to take this genome sequence data and apply molecular evolutionary analysis techniques to it to figure out where this bug had come from and how it was evolving. So they created a wiki site. It was basically an open lab notebook that was open to everybody in the world. They could check out the analysis that this group was doing, and they worked 24/7 on analyzing the genome sequence data and figuring out what was going on with swine flu. As a matter of fact, that very same day, that group working together as a team had figured out the origins of H1N1 influenza. They realized that this virus was made up of 6 parts of a pig virus that had been circulating in North America for a while and 2 parts of a pig virus that had been circulating in Europe and Asia. Only 5 days after the first genome sequence was made available, we had the first paper, official science research paper, published on H1N1. This was, again, about the origins of the virus. It was a different group from the wiki group, but they arrived at the same conclusions. And this is absolutely remarkable, to go from a genome sequence and 5 days later having a peer-reviewed and published paper using that genome sequence. It's the fastest turnaround we've ever seen in science. By about a week and a half later, you had genomic data available for about 69 different viral isolates, and the wiki group used this to do a molecular clock analysis where they calculated that the virus would have entered the human population probably towards late 2008 or early 2009. And this molecular clock analysis was included in really the first major paper about H1N1 that was submitted to the journal Science on May 5th, and in only 6 days it had been peer-reviewed, accepted, and made available online. Again, remarkable turnaround. And a month after that, by the time the pandemic was declared on June 11th, you had over 250 papers available on H1N1. If we go back and compare this to SARS, which was our last big outbreak, you'll see just how quickly we've managed to improve our response times. So SARS, we thought we were pretty fast with SARS. If you take day 0 as the day that we isolated the SARS coronavirus, it took us 19 days to get to a viral genome. And I will give a gratuitous plug here. That viral genome was done at the BC Centre for Disease Control with researchers from the Genome Sciences Centre. In 19 days, as I can't take any credit, give applause to Bob, though. Good job, Bob. It's the reason we're all still here and not dead of respiratory illness. In 19 days of H1N1, we had over 100 viral genomes. We knew where and when and how this virus had emerged. There were multiple papers that had already been published or were in press, and we were already growing up a seed strain for use in the vaccine. So, we thought we were fast in 2003, and we're really fast now. So how is this possible? How are we so fast at responding to this? Really boils down to 2 things: technological advances and a shift in the attitude of the science community. Technology-wise, it's never been easier, cheaper, or faster to sequence a genome. Back when the Human Genome Project started in 1990, it took 10 years to get to a draft version of one human genome. It took another 3 years to complete it, though many would argue it's still not actually complete. It cost $3 billion and took hundreds, if not thousands, of people. Fast forward to spring of this year, it took this guy 4 weeks, $48,000, and 2 of his friends to sequence his own genome in its entirety. The advances in this field have been absolutely remarkable, and they just keep coming. I mean, it's getting to the point where probably within the next 2 years, we're going to have a $1,000 human genome available within a week. Similarly, it's never been easier, cheaper, or faster to push data around the world. We've got things like email, collaborative workspaces like Google Docs and Wiki, and we've just got the network infrastructure to support all this. I mean, when I first got on the internet, and I won't tell you when that was, it was a long time ago, I would hit the get messages button on my email program. I would go downstairs and make a sandwich, and then I would hope that by the time I'd finished making that sandwich and came back upstairs, my 6 email messages would have downloaded, and sometimes they hadn't. Similarly, along with these technological advances, we've had real advances in scientists' attitudes, and you're seeing the ranks of academia and research populated by people of my generation, and for lack of a better word, I'll call us the file-sharing generation. So we were people that grew up trading our Commodore 64 games back and forth with each other. Totally dating myself. I look young, but I'm older than you'd think. We trade our Commodore 64 games, we go on BBSs, and we were the first people to use Napster and create Friendster accounts and things like that. And this really engendered in our generation this belief that information, and particularly knowledge, wants to be free and should be free, and that there should be no barriers to accessing it. And this has really manifested itself in research as an open access, and really the adoption of an open access mentality. Another gratuitous plug is that it's Open Access Week. In 2 weeks, UBC's got a lot of excellent events planned, so do check them out. Back when I was starting graduate school, PLOS, the Public Library of Science, and Biomed Central, both open access publishers, these were just fledgling concepts. They didn't exist yet. But now, just really about a decade later, these have become 2 of the biggest publishing forces in science and publish some of the most impactful journals that are out there. And in fact, a survey that was done earlier this year says that 85% of scientists support open access. So in addition to the spirit of wanting information to be free, scientists these days are also really big on collaboration. We appreciate that, 2 heads are better than 1, but 20 heads are better than 2, especially when those 20 heads all come from different disciplines and bring different skill sets. And I think this is really clearly illustrated when you look at how authorship of papers has changed over the last 50 years. So I have on the left a screenshot of Nature taken from an issue published exactly 50 years ago today, and you can see that there's 2 articles on this page whose authorship you can see. They're both single author papers, and it's kind of hard to see the references, but they all refer to single or dual author papers. On the other hand, you've got a paper taken from Nature, the issue that came out on Thursday, and this is much more typical of what you're seeing these days, and this is a paper with 36 authors. So we've really embraced collaboration and team science. Science has moved away from the domain of the individual and moved into the domain of the community.
I'm going to have to stop her because we're starting to run out of time. But Jennifer's done a very good job there being a warm-up act for what I'm going to tell you about in the last 10 minutes or so. And that is our experiences over the summer with a condition caused by sugar toxin-producing E. coli, or STEC. So hemolytic uremic syndrome is caused by this kind of E. coli. You get bloody diarrhea, damage to the kidneys and brain, anemia, loss of platelets. It's all down to this. This is the sugar toxin. So AB5 toxin has got an active subunit and then 5 identical B subunits. Now, in the summer of this year, May and July of this year, through to July this year, there was an outbreak centred on Germany, which you probably heard about. You must have heard about. It was in the news, all over the news. Over 4,000 cases, over 40 deaths. It was linked to sprouting seeds, the fenugreek seeds, in fact, characterised by a very high risk of hemolytic uremic syndrome. Up to 25% of cases getting hemolytic uremic syndrome, which is much higher than usual. Females particularly at risk, and you can see mainly centred on northern Germany, but actually scattered also through the rest of Germany as well. And this just shows you the epidemic curve of that particular outbreak. You can see the worst of it was around May the 21st, 22nd here. And this shows you both the gastroenteritis and then those specific cases that got hemolytic uremic syndrome. And you can see just by looking at this, females particularly struck by hemolytic uremic syndrome. We really don't have an answer as to why females suffered so badly, both in terms of incidence of disease and the severity of disease. One perhaps controversial explanation might be that real men don't eat bean sprouts, and perhaps it was the females that were eating the salad while the men were eating the barbecue and the steak. I don't know. Anyway, at the heart of this outbreak was this guy here working in Hamburg, Herr Dr. Holger Roeder, and he's a microbiologist. He's not a genome specialist, and he was kind of thinking, what was he going to do with the isolates? He had lots of isolates. What was he going to do to actually find out what this strain was about? Now, people of my generation know what you do when you're in trouble like this. You call international rescue. And I think probably the cultural illusions probably just about persist in your generation. You know what comes next. If you call international rescue, what happens?
5, 4, 3, 2, 1. Thunderbirds are go.
Well, it wasn't quite that dramatic, but he did actually call on international help, and the international community responded in a very dramatic way. Started off where one of his Chinese postdocs said, well, why don't you actually send the isolate to BGI Shenzhen, which is a genomics institute in China. It's in fact now the biggest genomics institute in the world. And so that's what they did. It didn't go in Thunderbird 2, it went by conventional air freight, but they sent the DNA to China. And BGI had just taken receipt of one of these instruments and decided to genome sequence the isolate on this here, the so-called iron torrent personal genome machine. This is a new so-called benchtop genome sequencer. It's about the size of a laser printer. It has millions of wells in there which read the sequences, and underneath the wells is a microchip which actually acts as a kind of massively parallel pH meter detecting the release of protons during the sequencing reactions. So this also has some very attractive features, about a 3-hour run time and only about £500 cost per run. So very cheap and straightforward. In fact, as it happened, a couple of months before the outbreak, a few months before the outbreak kicked off, I'd actually won our own iron torrent personal genome machine for use here in Birmingham. This was a European-wide competition that Iron Torrent introduced when they wanted to introduce the technology in Europe, and I was one of 250 applicants. I was one of the winning one, two winning ones out of 250. And what that meant was that we were actually ready to analyse the data. So what BGI did was they actually released their Iron Torrent data. Pretty much nobody else in the world was actually looking at Iron Torrent data for these kind of bacterial applications. But my bioinformatician here, Nick Lohman, happened to be at a conference and saw the data and said, oh, why don't we actually do something with this? So he knew how to analyse that data, to assemble the data into a genome, and then he released his assembly and said to other people, why don't you come and help? Why don't we crowdsource the analysis? And that unleashed a wave of creativity and energy. Within 24 hours, he'd assembled the genome, but within 2 days of release, it had been assigned to an existing lineage. Within 5 days, diagnostic tests had been devised. And within a week, over 24 reports had been filed on this open source wiki. So this is the first thing that came out of those analyses was this guy here, Mike Feldgarten, who blogs under the name of Mike the Mad Biologist, and he is mad in both the UK sense and because he's a bit crazy, but also in the US sense of always been a bit angry about the world. But he actually, in a very sober moment, said, well, hang on, this new strain that everyone's saying is new isn't not that new, because if we look carefully at the molecular signatures within the genome, we can see that it is actually related to something that happened, to a strain that was isolated 10 years before in Germany. And as I say, within a week we had 24 entries, and you can see this is just a screen dump of the entries that were on that open source wiki, people communicating using Twitter, blogging on what they found as well. You can see the international nature of the collaboration just from the names there. We've got Spanish names, we've got Chinese names, we've got Muslim names, we've got English names, Polish names, All of these people chipping in together to help analyse the data. And this actually caught the attention of the news media and even the conventional scientific publishing companies. So we got into Science here, into Nature on the news blog. Other bloggers took it up as well. What were the messages that were learnt from those analyses? Well, the main thing was that these pathogens don't bother with passports. It was not a new strain, as we said, something seen in Germany 10 years before. But surprisingly, another close relative was seen in Korea. And when it came to the genome sequence and comparing that with other genome sequences, the closest genome sequence strain was actually isolated in the Central African Republic in the 1990s. It became clear that this is actually a lineage that was circulating in human populations, so-called enteraggregative E. coli lineage, which was a bit unexpected. Normally when you get hemolytic uremic syndrome, you're kind of looking for where's that cow shit got into the human food chain. Basically, either it's cow fecal contents have actually got into beef, or vegetables have been contaminated in the field or something. But here it was a lineage that seemed to spread from human to human. It's clear that bacteria evolve very quickly. We saw virulence factors jumping around from one lineage to another. In fact, this strain was antibiotic resistant, even though nobody was using antibiotics. We don't usually use antibiotics to treat diarrheal infections, and certainly we avoid them in hemolytic uremic syndrome because they're thought to make it worse. And clearly, it's still infection's still a problem. Emerging infections that just come out of nowhere can still present a problem and a threat even to the most advanced societies. So, Germany is one of the most advanced societies, one of the richest societies on the planet, but here it was devastated by infection. Following on Jennifer's points, I mean, what we actually engaged in here, we call it open source genomics, where there was this confluence of the high throughput genomics, being able to sequence things very quickly, liberal approach to data release, and then crowdsourcing of the analyses. And we established really that social media, things like blogging and Twitter, instead of just being a complete waste of time. I mean, I was initially dismissive when Nick told me he was doing these kind of stuff. These actually can powerfully augment the usual channels of academic discourse. Does this mean that science is going to be changed forever? Well, perhaps not in ordinary science, but for public health emergencies, I think this is going to be the way forward. We have these benchtop sequencing platforms now that allow us to sequence genomes very quickly, and one of the key themes of my own research at the moment is to try and push this into clinical practice so that we can actually just sequence things quickly enough to have an impact on patient care. We actually managed to square the circle of not just putting all this stuff into blogs and Twitter and so forth, but we actually managed to get a paper, conventional scientific paper in a conventional scientific journal published on the back of this. And not just that, we got it into the highest impact journal of all, the New England Journal of Medicine. We got a brief report there, came out earlier this year. Here's us celebrating with our German collaborators, and this is Dave Rasko, who's a Canadian who worked also on this E. coli. In fact, there were 3 other efforts, genome sequencing. His was one of them, and we managed to take an opportunity to have a glass of champagne with him because he was published back-to-back with us in New England Journal. And that's me finished. I've put in Jennifer Gardy's slides into the set of slides, the PowerPoint slide that's been uploaded onto SlideShare. I'm now happy to take any questions.